This afternoon with Dr. Tammy Ivanco from two departments, the Department of Psychology on this campus and the Department of Human Anatomy and Cell Sciences on the Bannatyne campus downtown, where health research is conducted. And Tammy will talk to us today about the phenomenon of plasticity in the brain. That is, how does your brain change with experience when you learn something new, whether it's an academic matter or a skill of some sort, but also plasticity in the brain when the brain is injured. One thing we've learned over the last decade or so is that it used to be thought that when there was some kind of a brain injury, well, that was it. It was permanent. But we now know a great deal more about how the brain is able to assume the functions of a damaged part of the brain. And the brain is, that is to say, the brain is plastic. That is, it can adapt to injury and to all kinds of different things. And I will add that there is a large push on at this university in the general field of neuroscience. We're hiring 7 faculty members at the Bannatyne campus, primarily along the lines of neuroimaging, whereby we can, a surgeon, for example, can have an intraoperative image of the brain tumor that he or she is removing and can actually see the margins and get the entire tumor removed, whereas that was not possible prior to this kind of technology. So there's a lot of emphasis being placed at this university on neuroscience in general, and I think Dr. Ivanco will be able to whet your appetite a little bit with respect to this research area. So thank you, Dr. Ivanco.
Am I okay to start? Okay. Thank you very much for the introduction. I appreciate it. And I appreciate the chance to get to talk to you today. As Dr. Glavin suggested, I was going to talk about some things about plasticity as well as looking at a broken brain. Just to give you a bit of an overview on what I'm going to talk about. The purpose of this is to give you some idea of what's happening in the future research of neuroscience as well. And so what I'll do is I'll start off talking a little bit about what a broken brain is. I'll tell you sort of some general things that we're doing in my lab and where I see the future with respect to some of the things that we're doing, but also give you a feel for some things that people are just starting to look at that you may actually be bigger players in the future. And so I'm hoping I can also give you a feel for some of the cool and exciting things you could do in the future and maybe have some colleagues from this room. So we have to start off talking about how we mend a broken brain. We need to know about what it is to have a broken brain. And so many of you have probably heard of things like this. Stroke is a very common injury. It tends to be something that we actually hear about with older individuals, but it's becoming more prominent as we have more stressful lives. We're seeing younger and younger people having stroke. The way that we eat, the way our lifestyles are, our sedentary lifestyle, sitting at a computer, is not necessarily the best thing for our brain. We also have other injuries. It turns out human beings are kind of a violent creature. And so we have injuries like gunshot wounds, traumatic brain injuries related to bombs, and other kinds of things that really impact the brain by going into the cavity in your skull and injuring the brain. The other thing that we're hearing a lot more about these days are injuries related to sports, like concussions. Hockey players, football players, we're hearing a lot about how they have repeated brain injuries, and yet there are things that we don't typically talk about as brain injuries because, well, they fell down, they got tackled, they hit the ground. You know, yes, they were a little bit dazed when they got up, but they got up and they played again. Those kinds of injuries are sort of our invisible injuries, where we actually have repeated injuries happening, and we only know about them when there's something serious that happens, like a young kid getting a concussion that's actually maybe his 10th concussion, and dying after that type of injury. So we need to know a little bit about brain injury related to that as well. And then tumors, something that we're always concerned about with brains, because tumors are very difficult to remove from brains. And as we have an older population, and our population is only getting older, we have to worry about things like diseases like Alzheimer's, potentially things like Parkinson's disease, perhaps things you've heard about. The other thing that's, again, an invisible injury is damage that happens in utero. So when a pregnant woman actually has a fetus that is injured in some way. So what I'm going to do today is talk about a couple different kinds of injuries and some things that we're doing and some directions. So this is sort of related to brain injury like concussions. It turns out that one of our major sources of brain injury are vehicles and accidents. And we think about the idea, well, you can be in a car accident and get out of the car and walk around and you're fine. That's not really how it works, because there's lots of things that can happen inside your skull. We have this great protective coating on our skull that's supposed to protect our brain, but it turns out we can actually damage our brain inside the skull simply because it's floating around in some fluid. It's kind of like jello in a big balloon. So when your skull gets banged around, your brain gets banged around as well. I want to show you some things that happen to a cell. So if you watch this cell, see an injury, and what we see is that the cell actually changes. It loses part of its connection. See if I can play that quick again. See, the cell is functioning, looking good. All of a sudden, bang, there's an injury, and then the cell breaks. That's a really bad thing. The yellow in the cell actually changing, it means that the cell's died. So we might actually look like we're okay, but have a pretty serious problem with some of the cells in our brain. So we need to actually think about what's going on with that as well. Alzheimer's disease, this is a pretty obvious change in the brain. If you look at the normal side on the right and the Alzheimer's brain on the, the normal on the left, Alzheimer's on the right, other left for me, we can see that the brains look quite a bit different in this case. So how do we actually fix a brain when there's no cells or the cells have changed? And how do we fix it when the connections are all changed because of an injury like that? Again, I talked about stroke. Stroke is essentially a lack of oxygen, and stroke can actually occur in a number of different ways. One is where you have a blockage. So this is sort of like you eat French fries too much. You have your cholesterol, you have blockages in your arteries, and you stop blood flow. This is really bad for your brain because your brain doesn't store any energy. And so when it loses blood, essentially it has no oxygen and no source of energy. So this is really bad for the brain. The picture on the bottom corner shows a brain scan and an area of the brain that's actually essentially inactive. And it's dying for the simple reason that it hasn't had anything like oxygen. Now, Dr. Glavin talked about how we're getting people in the imaging program, and that's great. But one of the things that's a real challenge is that with all those types of injuries that I just talked about, one of the assumptions is that we have imaging. We can solve all brain problems. We can understand the brain. The problem with imaging is that it's still pretty new. And so we can get some idea of what the brain looks like. We have some idea of what parts are active, but we're not really good at actually saying what happens at individual cells. So when it comes to injury, we've got some of these great tools that are sort of on the cusp of getting really good, like magnetic resonance imaging, where we can look at the brain in terms of a picture, but it doesn't tell us what's happening at individual cell levels. It doesn't tell us very much about the biochemical properties of the brain, and it doesn't tell us a number of things that we still need to know. One of the other things that happens when we talk about injury, and especially things like Alzheimer's disease and Parkinson's disease, and you probably have heard this term, is that, well, let's do research on embryonic stem cells. We can get embryonic stem cells, and we'll put them in the brain, and that'll fix it. And the idea is certainly a good one, but right now we don't have a lot of ability to take those cells and put them into the brain and have them be very functional and effective. So we have some other steps to take before those two tools are really good. So what I'd like to do is talk about some of the things that people are doing now to sort of set the stage for what we will be able to use later. And this is sort of where plasticity comes in. So what is plasticity? Well, it's the ability of the brain to change in response to experience. So we can learn things, and our brain will change. If I ask you to tell me about an experience, anyone got an experience that they have? Something exciting? Somebody's got to have something. Anybody go to Disneyland recently? Yeah. I'm sorry? Oh my gosh, that's a big experience. What about other things? Experience. You went to a conference today. Okay. What about another experience? Somebody over here. What's an experience? Sailing and capsizing. definitely an experience. So these are all things that are memorable. And hopefully conference is kind of memorable too. And that's an interesting way to talk about it, because when we talk about experiences and somebody asks you, tell me about an experience, you typically think about something that is a big deal, an event that you remember, and it tends to, more often than not, actually be positive rather than negative. And the individual who said about being in a car accident, that is a definite experience, but the two are often thought of as being very different. The interesting thing is that your brain doesn't treat them very different. Your brain is so interested in actually learning about the environment that it'll actually treat doing something like coming to a conference and learning something new in a very similar way to what it does when it actually is damaged. And so this is actually very useful because you can take tools like learning and get a little bit of information about how the brain changes. You can use models of damage or cases when there is damage and actually understand what the brain is doing when it isn't functioning normally. So it's very effective for research purposes. The other thing that happens to be fairly useful is that we don't have to necessarily study human beings to understand something about the brain. If you look at some of the information on what we know about the brain, it actually doesn't come from human beings. It comes from animals. And the reason for that is that animal brains have some of the basic structures that the human brain has. And this redundancy that we now know very well from things like Darwin's theory is very useful because we can actually understand something about an animal brain in a laboratory and then translate that into looking at human disorders as well. So this is very effective. So one of the things that I'll do is I'll talk a little bit about some of the things that we've learned from animals that we can relate to human beings as well. So one of the reasons that we can do some really neat things with animals is that when we look at the brain, they actually have a lot of the same processes that happen. So we can look at things like when cells are born, we can look at where they go, we can look at injury, and we can actually say what's happening in this brain. And then we can say, well, if we know this is happening in an animal like a rat, this is probably what's happening in a human being as well. And what I've shown here is just a scale to give you some idea of when we have a human and a rat, what time points are actually comparable. So a rat is a little bit interesting because a rat can actually be born and it's out in the environment and starting to learn things at a time point when in a human being that baby is actually still in utero. So a rat gives us a really good idea of what we can look at in early injury, and then as it gets older it can actually mimic injury in adults as well. So some of the research questions that can be asked are what happens with injury? Can aging change injury? And does timing of injury matter? And all of these are really interesting questions, and so people who are interested in neuroscience tend to be drawn to some of these questions because we want to know what's actually happening in the brain. And injury is a really good way to sort of look at mechanisms. If we can actually have an injury, we get some really good idea of what happens when the brain isn't quite working right, but also some of the mechanisms that are involved in learning and memory as well. On the left-hand side is a picture of a neuron, and this neuron is stained with something that allows the cell to be labeled very dark black. This particular stain is very useful because if you look at it, you can actually see that you can see a cell that is, it's got a cell body, this part that's in the center, and a lot of processes. And these processes on these cells end up being quite interesting. The other thing that is shown on this image is actually in the cartoon part of the image. And what they've done here is they've drawn out what the cell would look like. It's kind of a schematic, but they've given you all the processes that are on these cells. And so this is a very typical brain cell. And I'm just going to use a pointer on the one slide here. So we've got a cell body that's drawn and processes coming off the cell body that are called dendrites. And they go up and they come off the cell body. And these particular processes actually collect information from other cells. And on the bottom of the cell is something called an axon. And the axon is what communicates with other cells. So this is kind of like the telephone line from this particular cell that talks to other cells. And then the receiving end is like what are these processes up here. Knowing something about these cells gives us a really good way to actually look at what's happening with injury and with the brain in general. So one of the things I want to do is I want to talk about this little box that's up here that's blown up in this image here. What we have is a cell that has a process, that's this part, and an axon, which is this part down here, and they're actually touching. One of the things that we do sometimes when we actually want to evaluate learning is count how many of these little connections there actually are. And if you look at this image, you see it's a little fuzzy. Every one of those little fuzzy parts on that cell are these connections. Now, the connections can actually just be counted, and we can get an assessment of how well the cells are communicating with each other. But we can also do something else, and that's actually look at what the cells are doing in terms of what that connection looks like. So we're going to play a little game here, and you've got neighbors. Almost every one of you has a neighbor. And so one of the things that happens with a cell that is just making new connections is it makes something called an immature connection. An immature connection looks like this. Everybody do this? And it's navigating the environment. And what it's doing is it's going to look for a neighbor to touch. And it's going to just touch its neighbor. Okay, so we've done something and we have a connection. And so you could imagine that a cell is doing it by a chemical means, but if you just gave a little tap with your finger, the effectiveness of that communication is fairly small. Would you agree? Just touch your neighbor. Okay, the next part of this exercise requires constraint. A very effective synapse, which is like what's shown in here, this connection between two cells, looks like this. Okay, so this is where constraint is important. You're going to navigate and touch your neighbor. Now, you can see how the second one is more efficient. So sometimes what we want to do is count. Sometimes we want to look at what those synapses look like. And sometimes what we want to do is actually change these kinds of connections into these kinds of connections. And so with brain injury, when cells are growing and changing in response to the injury, we want to make sure that we can actually make a more solid connection. One of the things that you may not be aware of is that children that are born with mental retardation like Down syndrome have brain connections like what we just talked about with the finger. We want to try and get them to have brain connections like that fist, where they're very effective communicators. One of the other things that happens with the cell is this section here all grows. And it can grow bigger and thicker, or it can actually be pruned as well, so it gets a little smaller. So you might imagine how many of those fingertips or fists could actually touch a cell. And that actually influences how much information gets transmitted to that cell as well. So this is very important when it comes to injury, because we want to have the most efficient communication system possible. But if we use something like MRI, we can't see what's going on here. So this can grow if we actually teach. I have a thing that I do with my rats that I teach them to reach through a slot. And Mr. Dentry might have heard about this, where we have rats that reach through a slot and they get food pellets. And when they get food pellets, they do this, they grow. This is very interesting. So we know if we teach them something, these cells will grow. So you might assume that if we actually have kids with mental retardation or after injury, we might actually enhance growth by just teaching them how to do things. The other thing that happens sometimes with injury occurs with this. And that little video clip I showed early on, we saw an impact that is representing a car accident perhaps, and a brain injury, and the axon broke, so this section actually became unattached. One of the things that we want to know when we have injury is what's going on with those axons as well. So we're going to do another demonstration. You guys have all been to football games or baseball games, that kind of stuff? You know what the wave is? Can we start it on this side? Woo! So you guys are great axons. You guys can communicate. So if I have a signal coming here that I want to send that way, we're really good at this. Woohoo! Now what we're going to assume is that this part right here is a break in the axon. We've had damage. It's broken. But the other cell is way over there that I communicate with. So this part can't actually follow. So if we do the wave, it's going to stop right here. That means that section doesn't communicate. A couple axons will, but not everything. And actually, the people who put up your hands, you're actually very effective, because when we have an injury, it's not all axons that are broken, but some are. So you kind of get the idea that when we're talking about communication, we have a few things we need to look at. Okay, so we've seen what happens with axons and with dendrites and connections. So I just want to tell you a little bit about how my lab's been looking at those.
So the next animation or video we're going to see is in fact two axons from the eye coursing across the brain to find their eventual visual system targets. And you can see the growth change. This is from work by Christine Holt and Bill Harris. And you can see they're leaving a trail of an axon in their wake.
And when we have learning, we can actually see things like those projections increasing in number or decreasing in number, depending on what kind of learning we're actually doing. Sometimes we actually want to have fewer connections with other cells, and so our brain is actually very good with those kinds of connections. So this is just another picture of a cell. So I mentioned that you can use animal models to look at injury, and one of the things that my lab's been doing is actually looking at rats and creating injuries in baby rats. And what we do is we try and mimic what happens in a stroke, where we have a blood clot that forms in the brain and causes a lack of oxygen. And what I've got here are just pictures of a rat brain where I've taken little slices through the skull or through this, the tissue like this, and they've been put on slides. And what you can see is that the rat brain here has a nice big hole in it. So we can actually very effectively mimic what a rat brain lesion, or sorry, a human lesion in a rat, and create a hole. And then we can actually look at all those things that I just told you about. We can look at connections between cells. We can look at what the dendrite looks like. We can look at how axons actually communicate with other cells. And we can learn how effective the cells that continue to exist are, which ends up being really useful, because if we have people that have stroke and injury, we want to know, are their cells effective, and can we do anything to rehabilitate them after that fact? So one of the ways that we do this is we draw cells from the brain. And this is just a schematic of what one of those cells might look like. And we're interested in, do they actually get bigger? And sometimes we actually see these cells grow like this little red line or that little red line. And in order to actually look at these, one of the things that we're doing is a very low-tech method for counting how big this cell is and measuring it. And so one of the things that we do is we place these concentric rings around the cell, and we count how many times, oops, there's one more dendrite there. We count how many times this dendrite actually intersects with these circles. And if we have a known distance between the circles, we can actually estimate how much dendrite is there, how much a cell might grow or shrink over time. The other thing that we can do is shown in the next picture. And again, these are fairly low-tech methods. We can actually look at how complex a cell is. So I said that these dendrites can grow and branch out and make more connections. So the more complex they are, the more connections they can make. And again, we can have changes after injury, but we also want to look at this for rehabilitation purposes. And so this just shows when you have a cell bifurcate or divide in two, it actually has a greater complexity. And a lot of the analysis for looking at dendrites actually comes from analysis of trees. And so we talk about it as being a dendritic tree analysis. And we can evaluate after injury how complex a cell is. And then we could introduce a rehabilitation strategy and say, has it actually changed to become more like a dendrite from somebody who hasn't had an injury? So I haven't presented a lot of data. Oh, this is just a high-tech way where we can do it in 3 dimensions. I haven't presented a lot of data, but what I'd like to tell you is that in the rat model, in that tissue that I just showed you, injury does cause changes in cells. What we can find is that there's changes in the neurons. They actually change their shape, but there's variability in this. And it's injury dependent. So if we actually make the injury at different points in time, we actually see differences in how the cells respond. So what's needed then? Well, we need some real-time information. We need to actually be able to look at what's going on in the brain at all times. We can't just look at sections. We need to actually have more information. One of my other questions when I gave the questions earlier was, does aging change the brain? And it turns out that if you have injury early and you let an animal age, which happens with premature birth, for example, that what happens is that the brain actually looks different as it gets older. And so aging is something that normally taxes our plasticity system. And so if you have an injury early on, you've already taxed the system, so then you introduce another challenge, and the brain doesn't respond very well. It doesn't do well. And so one of the things that we need to do is think about, well, how can we actually enhance plasticity? How can we make axons repair themselves? So if we did the wave, that it actually went the whole way. Or if we actually looked at dendrites and connections, we had more of those fist-like connections. And one of the places where we're thinking that this might actually be a place to go, or sorry, a place to go with this is something like stem cells. Can we actually put stem cells in and have axons grow across an injury? Right now, we're not there. We can't actually make this happen very effectively, but it's certainly a place where a lot of research is starting to happen. And then timing matters. If you have an injury happen in a really young brain, sometimes that really young brain is actually very effective at basically creating new pathways to allow for learning. So if an injury happens to a baby, quite often it looks like they actually recover completely from it because they develop new strategies and new brain pathways to deal with some of the same kinds of environment challenges that they have and learn slightly differently, but they still are able to do it. One of the things that is still a bit of an issue is that we still need some more real-time information. What's actually happening all the time? But these kinds of things, like real-time information of what's going on in a neuron, is still challenging. There are some people actually on this campus who have been looking at how to make the magnetic resonance imaging have better resolution. And with techniques like that, we'll be able to actually look at what's going on with individual neurons. How are they growing? Do they respond to rehabilitation? Do they respond to drugs? Can we actually make them grow into something that they look like before an injury, and so have the best outcome after an injury? So this is sort of just a bit of what we need still. We need people who do MRI research. We need people who actually take MRI from just looking at blood flow, which it does right now, to actually looking at what happens at individual neurons. The challenge is that this is a technology that is pretty new. And so it's going to require engineers, physicists, cell biologists, neuroscientists, people who actually have supercomputing skills. So if you're looking for places to go, all of these kinds of fields have really good opportunities in the future to really expand what we know about what's going on in the brain after injury. Stem cells, I said, is a potential area for looking at for rehabilitation. Stem cells are still pretty new in their research milieu as well. We have a lot of people who have been looking at how to keep stem cells alive, how to actually change them into different kinds of cells. But we don't have very many people actually taking stem cells out of a dish and putting them into a human brain. We don't have very many people actually doing that and putting them into a rat brain. And the reason is that when they get put into a rat brain, we don't necessarily know all the cues that allow them to grow but not become tumors. We don't know all the chemical signals that allow them to grow and connect effectively so that they basically fill in a hole and act like the normal tissue. They tend to do something that's a little bit different. So we need people that do chemistry and cell biologists and neuroscientists to actually look at stem cells and how we might have put them into the brain. This is a place where we need research in the near future, and some of our greatest advances will actually be in the near future, I think. Now, I want to talk about a couple different kinds of other injuries other than, say, lesions to the brain. And what I want to talk about is what happens with an early injury and why that actually might be interesting to look at as well. And we have a number of different kinds of injuries that are actually getting a bit more attention in Canada as well. And the one I want to talk about today is autism. And autism is a disorder that I don't think was getting a lot of attention until recently. And Canada actually set up a national strategy for autism, or at least they tried to. And they brought together a bunch of people to look at autism and try and figure out ways that we could actually put money together and direct some research at autism. And part of the reason is that autism seems to actually be a disorder where we actually see increases in number of kids that are diagnosed with this disorder. But it's also a disorder that is a little bit invisible in terms of what the brain problem is with autism. We think that with autism that some of those cells that I was talking about, all those dendrites, are actually becoming more numerous, and there's more of them. There's more connections, not necessarily fist-type connections, but the smaller connections. And that kids with autism actually have too many connections to make sense of the world. And what I mean by that is that if you were in a room with a lot of noise and you were trying to figure out a conversation that was happening across the room from you, you'd have a hard time. If you were in a room with the same number of people but there's only one conversation happening, you'd be able to hear that. And what happens with cells is that all those connections that are made with other cells basically create noise. And the best way that we actually have cells communicate is when the noise has a consistent signal. And we talk about that in engineering terms as having good signal-to-noise ratio, where the signal is amplified above the noise. What we believe is happening in autism is that that signal is just a little too much, that it's actually not consistent, it's not synchronous, and that we have kids have a hard time actually making sense of the signals that are coming in. Just some general data on autism. We have about 1 in 1,000 kids that are actually diagnosed with autism. It happens very early on. We diagnose kids at about 3 years of age, but the actual theory on what's happening is that brain injury is happening in these kids much earlier on. And we know that kids, you've probably heard about this, where kids with autism are diagnosed as essentially not being able to communicate or having started language skills and then regress back to not communicating. And they have a couple other developmental milestones that they don't meet in terms of not being very good socially. So what happens with autism? Well, this is a really confusing thing because it's not just one gene that creates autism. That would be really simple to understand it. It's actually multiple genes. And so the problem is if it's multiple genes, we also don't know how those genes interact. We know that environment matters. And we actually have some evidence that some environments actually have risk for creating cases of autism. And I'm not talking about vaccines here. There's no evidence whatsoever that vaccines create autism. But we do know that if you actually look at mothers who take epileptic drugs to control seizures during pregnancy, if they take them at a very specific time, they do have kids that have autism. And we also know that if they take these drugs at slightly different times, they can actually have other body abnormalities in their children as well. The problem with autism is that there's no single brain region that looks like it's damaged. So right now we don't know very much about it, but one of the big goals is understanding it. I mentioned that we have this idea that it's a really early brain injury, and one of the reasons we believe that is that our brain is actually sort of formed evolutionarily from the inside out, and some of the oldest structures are on the inside of the brain and in the brainstem. And it turns out that these areas are also things that are created early on in our development, in utero. And so some of the places where we've seen cells that are not necessarily growing right, or clusters of cells that don't form correctly, is in this brainstem region, this very old area of the brain that actually develops really early on in utero. And this is just an image showing some of the other body problems that can actually occur in individuals who take drugs like an anti-seizure medication or a drug called thalidomide. And most of you probably don't know what thalidomide is, but thalidomide was actually given to pregnant moms as something to make them not be nauseous during their pregnancy. And it was thought to be safe because they gave thalidomide to rats, and rats were just fine. But in humans, when they gave it to pregnant moms, the offspring were developing and being born without limbs and other kinds of problems, like they were missing ears if they had it early on. They had thumbs with an extra joint if the moms got thalidomide very late. So this actually was very interesting because we could actually note that when children in utero were exposed to something like thalidomide or the anti-seizure medication at a very specific time, they had characteristics of autism. You might go, well, we don't look the same, and so we can't look at that. But it turns out that development in basically all creatures starts off fairly simple. Organisms that don't have very complex brains, things like chicks and birds, and then human beings actually look very similar. And so this injury that we think is actually happening that is exhibited in kids with autism essentially happens really early on, just before their brain is actually forming. And so we can actually use animal models to look at this as well. And this just shows early on, we see an embryo, a picture of an embryo. And what happens during development is that our spinal cord actually forms and folds over into a tube. And the formation of this tube ends up being really interesting. And many of you have probably heard that pregnant moms should take things like folic acid to ensure good neural tube development of their offspring. But it turns out that neural tube is very interesting when it comes to autism. And people have been looking at animal models and doing things like manipulating animals in utero by giving something like an anti-seizure medication when this neural tube is just forming, which is shown in this picture, in rats. And what they've done is they've actually been able to get really good models of something like autism. Now, we've been using this model in my lab and getting some really interesting data suggesting that the animals don't play the same, and they have a slightly different pattern of how they play. But the future of this kind of research is actually far beyond what we've been doing right now. And one of the things is actually looking at all the risk factors for autism. What actually is a risk factor for autism? And it turns out that autism is something that we see a lot more in kids that have other genetic problems. And so Fragile X is the leading inherited cause of mental retardation. And about 50% of kids that actually have this gene mutation have autism. And so this gives us some really interesting ideas about what a risk factor for autism is. And it probably is the case that we have a whole bunch of risk factors for autism, and we just don't know what they are yet. We have some other kinds of genetic disorders that also present with symptoms of autism. So some of the social problems, some of the language problems. So in terms of what's going on in early development that actually creates a brain injury, it presents a really neat place for people like yourselves to actually think about where you might like to go in terms of studying and doing research. And one of them might actually be into looking at genes as risk factors for disorders like autism or some of the other kinds of developmental disorders that we have. And so what we need are people who actually learn about cells and learn about genes and learn about manipulation of genes and are able to actually understand how genes and behavior are related. So this is sort of a summary of some of the things that I think are near future options for people who are doing science work. One of the other places where we have some research done where there's some other options is in the area of creating transgenic mice. And we kind of have a good set of animals right now where we've been able to knock out genes or have genes overexpress things. But I think some of the technology that will be available in the next few years for actually looking at manipulation of genes in mice is actually really going to lend us a lot more information about how the brain responds to having genes or not having them and having gene products like proteins turned off and on. And I think the nice thing about some of the newer technology for looking at mice as well is that these mice can actually have very specific parts of their brain targeted. So you can actually turn genes off after they've been born and look at things a lot more selectively than what we've been able to do in the past. And again, this is something where we've got some technology that's here, but I think in the very near future we're going to see a lot of changes in terms of how we look at genes in response to things like early developmental brain injury. So now I want to talk about going a little farther out in the future and what we might actually look at. So how much time do I have? I have about 10 minutes, right? Okay. And going forward sometimes means going back. And so what I've done here is I've told you that in the 18th century we had somebody who learned a lot about how it is that our brain signals to our body. And Galvani was walking through a market and saw that when it was lightning, that the frog legs in the market twitched. And what he did is he went home and he applied current to frog legs, and yes, he could make them twitch. And subsequent to this, people started looking at what electrical stimulation could actually do for us. And Sir Walter Penfield, who's actually a famous Canadian, who was at the Montreal Neurological Institute, became even more well known because he was interested in looking at epileptic seizures. But in order to actually help people with epilepsy, he wanted to actually remove brain regions that were contributing to seizures. And what he did is he used electrical current to basically touch the surface of the brain and get information about areas that were involved in the seizures. But also he used this stimulation technique to map out what parts of the brain were not involved in the seizures and get a really good idea of what should be spared. Sort of consequential to doing this, he found that he could actually map out areas of the brain, which ended up being very useful for people doing neuroscience. So we've had this history of looking at stimulation. We now have some really neat technology to look at stimulation. I want to show you a video that I think is just great, looking at some of the new stimulation.
I'm Roger Highfield. I'm the science editor of the Daily Telegraph. Now, words mean everything to me, but now I'm going to have the speech area of my brain turned off with a giant magnet.
A different way to stimulate.
Humpty Dumpty sat on a wall. Humpty Dumpty had a great fall. All the king's horses and all the king's men couldn't put Humpty together again. Humpty Dumpty sat on a wall. Humpty Dumpty had a great fall. All the king's horses and all the king's men couldn't put Humpty together again.
That may have looked like a strange party trick, but it's actually part of a serious research project conducted by Prof. Vince Walsh in a basement lab at the Institute of Cognitive Neuroscience in London. He's using electrical and magnetic brain stimulation to understand the most complex known object in the universe and how to treat conditions such as migraine and stroke. Just to show the power of the magnetic fields passing through my scalp, and that this wasn't a trick, this is what it did to our videotape, even though it was positioned a few meters away.
Star Trek. Yeah, very left eyebrow twitch.
It's hard to see because of the interference, but here I could still sing a nursery rhyme, even when he disabled my language centre, because singing depends on the other...
Too many things playing at once. Sorry about that. So what we've learned is that we can actually use magnetic fields, and it turns out that when you have a magnetic field, and many of you probably know this from taking physics classes, is that you actually can generate current. And what happens is this is localized in a very specific place next to that coil that you saw that he was putting near his head. What we've done in the recent past is actually use this technique to do things like turn brain regions off, like what we saw in the video here where the person trying to give the nursery rhyme couldn't actually generate the motor skills to get it out. And one of the things that, if we'd watched a little bit farther on, what he does is he sings the nursery rhyme and he's just fine because it's not necessarily associated with the same part of the brain. So techniques like this are being used for things like looking at depression, and the stimulation is being used to treat people with depression. And essentially it's being used because it can activate or inactivate brain regions. In the future, and I think in the more distant future, what we're going to see, and certainly the trends in neuroscience seem to suggest, is that people are going to use stimulation techniques like this to actually do things like change cognition. And so this is, again, one of those future technologies that we're going to see being developed over time, and probably in the next 10 or 15 years actually see some very interesting uses of these technology to understand the brain, to be able to turn off and on the brain, but also to help regenerate neurons that are damaged after something like stroke. One of the other places that we're seeing some interesting things happen that, again, are very, at the very early stages of even development, are doing things like using light to actually activate cells. And there was a recent Journal of Neuroscience paper that talked about this, where they're doing this in cell cultures, where they have cells growing in a dish, and they're shining lights on these cell cultures and actually causing cells to become active or inactive. The idea is that in the future, they will need technology that can actually have light transported to places within the skull and within the brain and cause cells to become active or inactive. Having cells become active might help with rehabilitation, because you can actually cause cells to grow, cause them to make more connections that are efficient. Having cells become inactive is actually kind of useful as well, because we have problems with something like epilepsy, where cells become overactive, and they become overactive too often, and they can actually cause injury as well. So this kind of technology is really just being developed, and in most part, it's actually an idea. And the image here is an artist's rendering of this, because we actually haven't been doing this except for in a couple labs in dishes. But the technology that would be necessary to do this is something that we're just sort of thinking about. How would we actually be able to shine a light into the brain and have it get to target cells? It's really a challenge. But if we can actually use something as non-invasive as light and get it to a target location and have cells become active, this would be a really good step for looking at rehabilitation, because we don't want to have situations where we go into the brain after an injury. One of the other things that I think is a place where we'll see a lot of advances in the future, we've been doing this, but I think people are starting to become more aware of the fact that there's a lot of chemicals in our environment that we don't use. But yet our body actually has receptors for these chemicals. And so one of the things that we do sometimes is smoke. And the reason that smoking is actually pleasurable to some people is that we have receptors in our brain that actually use the nicotine that's in cigarettes. But we now understand that we can actually go and look for chemicals that are in our environment and target things, like in the example I've got up here, things like tumors, and actually make it so tumors don't invade brain tissue, that they actually are something that are more easy to take out. So if we have somebody that actually has a brain tumor that's going to impact their cells and function, if we can give them a chemical and have that tumor not invade the tissue, it makes it something that's easier to take out and less likely to actually cause long-term damage. So again, I think these kinds of things are places where in the future we're going to have some really neat advances, because people are looking at some of these chemicals in very different ways. And one of the things, we talked about stem cells and the idea that somebody could get new cells and put them into your brain, but one of the things that we've learned is that the brain can actually create its own new cells. But we don't necessarily know how to turn that off and on. And so in terms of looking at long-term future goals, one of the goals is to actually make us turn cells on when we need it, and turn them on in the right place so that they actually grow and connect up and basically fill in holes themselves, or make the chemicals that are necessary for our brain to function effectively. So I think, again, this is a place where we'll see a lot of development in the future, and we need people who are interested in cell biology and have a good understanding of genetics and chemistry to be able to do this as well. I've got a video I'll show once I've answered some questions, but you might be going, well, you give me a whole lot of things that can actually hurt my brain. What can I do that help my brain? And we do know a lot of things right now that, again, I think we'll end up learning more as we do more research on this. But generally, to help your brain, eat well. McDonald's is not the best place to get your nutrition from. Sleep well. I think one of the things that's really challenging, and a lot of people report, is lack of sleep. And we know that it actually hurts your brain. It makes your brain not function well. If you are not good to your brain now, and you do things like eat poorly, you don't sleep well, you get sick, and you have an injury, you are basically setting up your brain to not respond very well. So the better off you are to your brain now, the better long-term outcome you'll have. And use your brain. It's pretty easy to sit and not do anything, but one of the best things you can do for your brain is to keep learning, exercise your mind and your body, learn new languages, learn new things, learn how to do different things that you don't do. Don't just stay at status quo, because in order to have a very healthy brain for the rest of your life and to basically deal with some of those things that I talked about as brain injuries, the best situation is to have a healthy brain now. So I think I've given some feedback about what future things are available for people like yourselves, and I'd love to try and answer any questions if you have them. The first video, that actually was an animation. It's not actually technology doing that. There is some images using a light microscope in culture dishes where they're doing things like damaging and they see the axons breaking. And there's some where they've actually got tissue and they've mechanically sheared the tissue so it shifts. And you'll see that in a light microscope image. But the video I showed was actually an animation. Oops, sorry. Okay, so the question was, what makes a younger brain better at repairing damage than an older brain? And what we think happens is that a younger brain is actually more plastic. As you get a little bit older, your brain actually sets up some of its connections in a hardwired fashion. It does it essentially to save energy. It learns things, it hardwires the connections, and then when you're experiencing some stimuli in your environment, it knows about those stimuli. A young brain is preparing to understand the world, and so it's a lot more plastic. It's able to actually change more easily than an old brain. But one of the challenges is making an old brain actually more plastic like a young brain. The Alzheimer's brain picture that I showed actually had lost cells. So the cells had died, and they actually got smaller as well. So on the one side was a normal brain, age matched, so it wasn't actually a young brain, it was the same age, but without Alzheimer's disease. So as you get older, one of the theories was that everybody actually had cell loss as they got older, and we now know that isn't true. But when you have Alzheimer's, you actually see cell death, and the brain shrinks. So you actually see a brain that has less volume than a normal brain. So it has fewer connections. And so some of the memory loss that we actually see with Alzheimer's disease is because cells have died. Does that answer your question? I'm not, but people are, yes. They do use human stem cells, but they also use rat stem cells. It tends to be more related to whether or not they would actually reject the cells. And so you're better off actually having cells that are similar to you, and you don't have as high rejection of that. And so when we look at human research, people want to actually use human stem cells to put into a human being. It's certainly been controversial for actually collecting things like embryonic stem cells. But in terms of rejection and the most efficient types of cells, it would be human stem cells. We're actually seeing them now coming from other locations, like skin even. What's going on in Parkinson's disease is actually there's a very small area. Remember I was talking about the brainstem area that's injured in autism? That particular area has a group of cells that create dopamine. And those cells that create it don't use it in that location. They actually send their axons. So remember we were talking about the fifth thing? They send their axons really far away and release dopamine into other locations. And it turns out that those axons actually go to motor cortex, which is right here, and a subcortical area inside your brain that actually is related to motor function. And so with Parkinson's disease, those cells die. And about the time you actually see about 90% of them dead, essentially you're not creating enough dopamine to have fluid movement any longer, and that's when you start seeing your symptoms of Parkinson's disease. So that's why it's a motor disorder.
Parkinson's patients also have there's another whole nervous system, a little miniature nervous system in your gastrointestinal tract, and Parkinson's patients have a far higher incidence of a certain gastrointestinal disease, which is related to the same chemicals that are necessary in patients who
Yes.
Many women do not know that they're pregnant for the first month or so, and during that time, they may very well continue to ingest whatever recreational drugs or what have you. So we know a little bit about. You've hinted at the fact that there are obviously vulnerable periods of time in gestation that are more vulnerable to insult than others. So, and during that first month, there's an awful lot of neurodevelopment going on. So where do you think we should go in that line of work?
Well, I think we're always going to have trouble knowing when a woman is pregnant. It's very hard to actually tell. And some people might care, but many don't. And so they're not looking for signals like that. And so I think this is part of the reason we have to worry about rehabilitation as opposed to doing something early on, because it's going to happen. We also have women who are at risk, like drug users, alcohol abusers, that kind of thing. And so those individuals, even though they might know they're pregnant, they're still going to consume substances that are harmful to the fetus. So we need to actually be able to do something that's rehabilitative ex utero, so we can target those kids who are born in that situation. But I think it's more rehabilitation than actually finding out what's going on. There are some suggestion that we might be able to do something early on if it's a genetic problem, but I think the substance abuse problems are not going to be targeted in the same way.
Please join me in thanking Dr. Ivanco.
Oh, thank you.
Thank you very much. Thank you very much.